more information now, starting with our next recipient, Dr. Peter Sicinski of Harvard University. He's going to receive the $500,000 Dr. John Trojanowski Memorial Grant. So if you would please come up and share your research with us. Peter, he didn't want to be called doctor, so Peter, there you go. Thank you.
Yes, thank you very much. I am told I'm not allowed to jump to the, like the previous speaker, although I was planning to. Thank you so much for being here, and most importantly, for your mission and for supporting our research. After having received this award, I familiarized myself with CART, and even closer. Okay. Okay. So thank you. After receiving this award, I familiarized myself with CART, and I am extremely impressed how profoundly important things you're doing for the community. You are supporting our research, but of course, you know, the ultimate goal is to help the people and the community. So I am profoundly grateful for your award, which means a lot to me. I am very humbled and very proud to be the recipient of this award, and my lab will do everything we can to make good use of this money. So I am originally not a neurobiologist. I come from the cancer field, if I can have a next slide. Right, so my lab is generally interested, or historically has been interested, in the molecular mechanisms that drive cell division. And we are interested in how at the molecular levels the cells divide, how normal cells divide, how cancer cells divide, and what are the differences between normal cells and cancer cells that we could use for cancer therapy. And this slide shows a group of proteins which we call cell cycle machinery. And those proteins operate in our cells, but they have been conserved. That is to say, they work the same way in our cells, and they work in several other organisms, including yeast. And the proteins that drive cell division are called cyclins, and they are represented here by okay, I'm not allowed to jump, so I'll just gesture by brown ovals. And the role of the cyclins is to bind to activate the enzymes called cyclin-dependent kinases, or CDKs. And then once bound, those enzymes put phosphate group on different proteins, and this actually drives cell division. Next one. For the purpose of today's talk, I will focus on the so-called D-type cyclins, and these are the proteins. The cyclins are called A, B, C, D, and I'm talking today about D-type cyclins, which bind and activate enzymes called CDK4 and CDK6. The next slide shows that the picture is actually a little bit more complicated. Yeah, because there are actually 3 D-type cyclins. It sounds like a detail, but it's going to be important later. There is cyclin D1, D2, and D3. And each of them can bind and activate CDK4 or CDK6. Next one. So through decades of work, we now realize that those proteins, D-type cyclins, are hyperactive in cancer cells. And this hyperactivation of D-type cyclins represents the driving force of uncontrolled tumor cell proliferation. Next one. So my lab has shown that those proteins are not required for normal division of normal cells, but they are essential in cancer cells, and in particular in breast cancer. So this is the experiment that my lab has done, where we took mice which have breast cancer, and then either on the right side, we genetically turn off those proteins in the whole mouse, including in cancer, or in the right-hand panel, on the left-hand panel, right here, we gave animals a small inhibitor, the chemical that inhibits this kinase. And there was no effect on mouse health. However, the cancer stopped growing. Next one. And based on this work and the work of several other groups, those compounds, small molecule inhibitors, enter clinical trials, and they represented a breakthrough in cancer research. So here we are looking at the data presented. Now I have to go for just for 1 minute, and I'll be back, I promise. So, because here we are looking at the progression-free survival of the women with metastatic breast cancer. And this is the progression-free survival of women treated with a standard therapy. And this is progression-free survival of women with the addition of those inhibitors. And as you can see, it doubles progression-free survival from about 10 months to about 24 months. So this really represented a breakthrough in cancer research, and now those inhibitors are being routinely used for breast cancer treatment. Next one. So these are the chemical structures of 3 CDK4/6 inhibitors that are routinely being used in breast cancer treatment, and they are now being used in clinical trials, over 300 clinical trials worldwide for the treatment of over 60 tumor types. Next one. Right, so now we understand that those proteins are hyperactive in cancer, and we know that inhibition, that giving compounds that blocks the activity of those proteins, has very profound anti-cancer effect. Next one. Those proteins are active in proliferating cells. On the right-hand panel, you see a breast cancer, and in black are dividing cancer cells, which have high level of those proteins. And here you are looking at the developing embryo, which also there is lots of proliferation. Next. But brain is composed of mostly non-proliferating cells, and therefore those proteins are generally not expressed in the brain. However, in the previous century, at the end of previous century, people have noticed that those proteins become occasionally expressed, not in normal brains, but in brains of Alzheimer's patients, and also in mouse models of Alzheimer's disease. But at that time, there were no specific inhibitors of those proteins, and therefore it was very difficult to assess whether this is just some additional effect, or there could be an important role in driving dementia. Next one. So these are our experiments. We are using mouse models of Alzheimer's disease, and these models have been engineered not by us, but by others to mimic the Alzheimer's disease. And we treat them for about 5 months with those FDA-approved, commercially available inhibitors, and then we wait 1 month. I'll tell you later why we waited 1 month. And then we test the memory using different tests. Next one. So this is the widely used test that we are using. We put mice into the water, and the mice don't like to be in the water. So we provide them with the escape platform, and we also give some visual cues so the mice can orient themselves. And then the healthy mice, after the few days, will remember where is the platform, and they would quickly find the one. But the mice with Alzheimer's disease are unable to learn, or they learn very slowly. Another version of this, we would teach the mice where is the platform and then move it to another place. And then the healthy mice with some rapidity will realize that the platform has been moved and would learn the new location. But the Alzheimer's mice are unable to do this. Next one. Yeah, so this is actually our experiment. And then, I still have to go back. I'm sorry, because I have to point out. So this is you are actually looking at our results, and you are looking how the mice are learning the presence of the platforms. The control mice, shown in black, are learning very quickly. The Alzheimer's mice, shown in red, are unable to learn, or they learn very slowly. And in blue are Alzheimer's mice treated with our inhibitor. They behave like wild-type mice. They behave like healthy mice. This one is the situation where we move the platform to another location. And then again, you are looking in black. These are control mice. They learn rapidly. In red are our Alzheimer's mice, which learn very, very, very slowly. And then they almost never learn as well as wild type. But in blue are the mice that are Alzheimer's mice, which were given our inhibitor. So this inhibitor nearly restored normal learning and memory. The next one. This is another test that we are using. Here we put the mice into the cage, and then we play a tone. And as we play the tone, we give mice a mild but unpleasant electric shock. And then, after a few times, when we put a healthy mouse and play the tone, the mouse remembers that something bad had happened and will freeze. But the Alzheimer mice do not remember, they would not freeze, or they freeze less. Next one. The another version of this is to put a mouse into a special cage, like a very bright colors. And then as soon as we put a mouse into this cage, we give her or him electric shock. And then again, the healthy mouse will remember that electric shock is coming and will freeze, and the diseased mouse will not. Next one. Okay, so this is our testing, and then I promise this is the last time I'm jumping, but I really have to show this. So this is the good freezing, rapid freezing of control mice. The mice with Alzheimer's do not freeze as well, but when we give mouse the compounds, the memory is restored, and now they remember, and they freeze with this. Next one. So I mentioned to you that we waited 1 month. Why we waited 1 month? Because we are treating mice with our facility, and then the testing is in another facility. And there is a mandatory quarantine. Quarantine means that the mice have to sit for 1 month, and we cannot access them. And I was fighting tooth and nail not to have this quarantine, because in cancer, these drugs work as long as we give them. When we stop giving them, then the cancer starts growing. So I thought that it's a waste of time to treat mice, because if we stop treating them, the memory loss will come back. But actually, I was wrong. And even after 1 month, we saw the effects of treatment. And therefore, we decided to push a little bit further and to treat mice and then wait 6 months. And after 6 months, 6 months is 25% of mouse lifespan, we test them again. And we found that even after 6 months break, the memory was partially restored. The next one. So another manifestation of Alzheimer's disease is the loss of those brain connections that you just heard about, them called synapses. So here in the upper panel, you see control mice, and just for our convenience, the synapse is marked by red dot. In the lower panel, you see the mice with Alzheimer, and they have those less dots. They have less synapses. But treatment with inhibitor restores normal number of synapses. It's shown here, and it's this quantification. We can see it immediately after treatment, or actually this picture is taken 7.5 months off drug, so meaning that for 7 months, these mice were not treated, but still the normal number of synapses persisted. Next one. So at the molecular level, we found that those proteins, actually the particular kind of CDK4, CDK6. I mentioned there are 3 D-type cycles, D1, D2, D3. This is D3 together with CDK6. That they are expressed in brain immunity cells called microglia, not in the neurons, but in microglia. Next one. And what we found is that in Alzheimer's, mouse models of Alzheimer's disease, those proteins are in higher levels than in control brains. Next one. And then we found that those proteins promote production of the synaptotoxic C1q proteins by brain microglia. So it was well established before we started our work that the microglia in Alzheimer's disease produce a bad protein called C1q. And this bad protein goes to the synapses, and it facilitates synapse loss. What we found, our kind of small contribution, was to find that in microglia, cyclin D3-CDK6 regulates this abnormal production of C1q by microglia, abnormal production of this bad protein. Next one. So you can see it here, and then we are actually staining. So you can see here the signal correspond to C1q and WT, which are control mice. And there is very little signal because there is very little of this protein. You can see 5x FAD. These are Alzheimer mice in a vehicle. These are the control mice, and you can see much higher level of this bad protein. And you can see in the last 2 panels that they're giving mice to different small molecule CDK4 inhibitors normalizes, decreases the production of those proteins. Next one. Next one is just quantification. Again, it's difficult without pointing, so maybe I'll just keep it and go to the next one. And then I mentioned that this C1q protein goes to synapses. So here, this slide illustrates that if we treat mice with the CDK4/6 inhibitors, then there is less localization of this protein to synapses. In the upper row, you see control mice, and we are staining here for synaptic markers, for proteins that is in synapse, and for C1q. So we're looking for co-localization, which is in those round circles. And you can see that in control mice, there is very little this BET protein going to synapse. In the lower left-hand panel, in Alzheimer mice, there is a lot of this protein going to synapse. This destroys the synapse. And the last two panels showing that when we give mice the inhibitor, we decrease the localization of this BET protein to the synapse. Next one. This is just the same message using another synaptic marker. And then again, you can see in the upper panel that there is very little this BET protein in the synapse in control mice. In the lower right, you can see that in Alzheimer mice, as other people discovered, this protein localizes to synapses. And again, our two inhibitors normalize this. Next one. And this is just quantification showing, well, again, it's difficult to point, so I'll just keep it. Yeah. Next. Okay, so this is our experimental plan. So we would like to answer 3 questions. The first one is that we propose that this is cyclin D3 CDK6 in microglia that is driving those pathological changes. But when we give the inhibitor to mice, the inhibitor reaches every cell in the body, or at least theoretically. So I want to make sure that this is truly the microglia. And to answer this question, what we want to do, next question, next slide, thank you. If we want to use mice that we generated to specifically turn off cyclin D3 CDK6 only in microglia, we have made mice that allow to turn off those proteins in any cell type, and we want to turn it off in microglia. And the hypothesis here to be tested, and this will have the same effect as giving mice the inhibitor. This is going to reduce the production of this the toxic protein, and at least partially alleviate the memory loss. Next one. The second question is that I mentioned that cyclin D3 CDK6 promotes production of this BET protein by the microglia. Next one. But we are not sure what it means molecularly. So we know that if we activate this kinase, the genes encoding C1q are more active. But how exactly does it work? What are the proteins between them? That we don't fully understand, and we want to understand it with your help. And the last one is that I think that the potentially most interesting outcome of the study is that the effects of CDK4/6 inhibitors we can see several months after the treatment has been terminated. And this is very exciting because it suggests that a time-defined treatment could have a I don't want to say curative, but a long-lasting effect several months after the treatment ends. So it cannot be so simple that you inhibit something and then you de-inhibit something. And we hypothesize that administration of those inhibitors profoundly changes microglia in a kind of irreversible fashion. Kind of in our lingo, we say it reprograms microglia. But we really want to understand it molecularly, and this is the last topic of our study. Next one. Yeah, so lastly, I want to thank you again for your support. As the previous speaker said, it's very difficult to get funding for different reasons. For us, it's because we are outside. My lab is well funded for cancer field, for cancer research, but not for neurobiology. So your funding will make this work possible, and without you, this work would not have been possible. So I'm really, truly, profoundly grateful for your help. Thank you very much.
He said, I'm sorry I jumped. I couldn't resist.
Yeah, I couldn't resist. I'm sorry, because for some of the slides, it's just impossible to understand them if I don't point.
Now, are there questions for Peter? Yes.
That's really a great question, and we had actually a discussion about this during the coffee break. And I'm in a cancer institute, so I thought it's easy to answer this, but I could not get the answer to this. And the reason being is that clinicians tell me that, first of all, the women of breast cancers are usually younger, so they don't have dementia. Number two, that when those drugs are in treatment relatively recently, and when there were clinical trials, people from dementia were not included into those trials. And thirdly, they tell me that with cancer patients, which is obviously such a stress, and also several treatments, like for example chemotherapy, that does affect the cognition. They say it's going to be very, very, very difficult to really dissect the effect of this, because precisely our hypothesis was, our hope was to test the notion that the patients with Alzheimer's who are getting the treatment are going to be progressing less slowly. But this data, neither confirming nor refuting, is not available. I think that, ultimately only clinical trials will tell whether this works only in mice, as many treatment in cancer, or whether this is something that can change also our lives.
Are you close to clinical trials?
No. Well, so we need to publish our work. So thanks to your support, we can complete it. And of course, our goal, our dream, is to have the clinical trial. But as you realize, there are several regulations in clinical trials. So it's going to be a while until such a trial is started. But that's, of course, our goal, I hope, our dream. Yeah.
Are there any known side effects to the inhibitor? I'm going to repeat for the streaming audience.
Yeah. Yes. So the inhibitors are generally well tolerated, but they do have they do have toxicities, and the toxicity is the toxicity is in the bone marrow, and some patients are anemic. And some patients are also having the low number of white blood cells. However, now the people are trying to develop next generation inhibitors. They do not inhibit CDK4 and CDK6, but only one of them. And also, not for Alzheimer's, but for brain metastasis. People are developing CDK4/6 inhibitors that preferentially go to the brain. That would be the best compound. The ideal compound with a compound that has a higher concentration. The current inhibitors, they cross blood-brain barrier, but the concentration in the brain is lower than plasma. The companies are trying to develop inhibitors that would preferentially go to the brain. That would be the best, because it would spare all the side effects that come from periphery. But despite those side effects, those compounds are relatively well tolerated. And recently I was in a conference. There's almost 1 million women that have been treated with those inhibitors.
Yes.
I have one small technical question I want to ask. So it's not for you guys, but it's my really strong personal interest as well. So I noticed your CDK inhibitor drug is very impressive on the EM you showed, where it rescues the synapse.
Right.
But I also noticed in your 3xTG mouse, there's hypomyelination, because you can see the myelin sheet is gone, basically, in the diseased mouse. But I think I see some shade of the myelin when they're treated with the drug. Do you think it can also rescue the hypomyelination in Alzheimer's disease?
It could. We didn't look, we didn't examine it, we didn't quantify this. We quantify the synapses, the number of synapses goes back. Also the morphology of synapses seems to be corrected, and also the number of dendritic spines is restored. But of course, you're right, there could be additional effects, yeah.
We can talk more after.
Sure.
I'm sorry, I wasn't going to try to repeat that one. beyond my capability. Any other
No,
questions?
it's a very good question.
Thank you. And we'll hear more from you in just a moment. I do want to say that I did mention that you are receiving the Dr. John Trojanowski Memorial Grant. He was a prominent academic research neuroscientist and former member of our CART review team. So each year, your grant recipient is done in his honor. He completed his MD and PhD degrees from Tufts University, neuropathy training at Massachusetts General Hospital and Harvard Medical School, and later rose to the rank of tenured full professor at the University of Pennsylvania School of Medicine. So we honor him every year with this particular award, and it's yours this year.
If I may add, actually, this is a classical Polish name. I come from Poland, and this is a classical Polish name. So the proper Polish pronunciation is not Trojanowski, but Trojanowski. So I looked him up last night because I thought he was born in Poland, but no, he was born in the United States, but probably his parents came. Yeah.
Thank you very much. So with that, we'll have Dr. Goforth and Carol Burdett present you the money.